Okay, so my name's Scott Wilbur. I'm at the University of Washington. I'm going to be the moderator for this user meeting, the virtual 2020 Astatine-211 user meeting. This meeting has been set up by the National Isotope Development Center, basically to inform interested people in astatine research, those that are already doing it, and some people that might be interested in learning more about it and potentially doing it in the future. In the past, there's been a real shortage of astatine for research, for applications. And one of the things that we've been involved with through the Department of Energy is production of astatine for sale through the NIDC. But clearly we're in the Northwest and a long ways from a lot of other sites. And with a 7-hour half-life, this is problematic with regards to delivering astatine. Other sites in the US can and are producing it now, and there's the Department of Energy is setting up or trying to set up a network of sites for production of astatine, I assume for both preclinical and ultimately maybe clinical studies. And we're part of that network. In today's talks, we have 4 sites, university sites that produce and conduct research with Astatine-211. We'd like to kind of bring things up to speed with regards to what's going on at these sites. And I guess I was a little bit of a hard nose in some regards. I required that everybody try to keep to a 10-minute limit in their presentations so that we could have time for discussion after those presentations. We've also had a later presentation that was requested from Global FIA and Graham Marshall, which he will present after the university presentations from a company's perspective of some things that are going on. We will have Q&A. There's 2 ways to ask questions while we're here, and I suggest that you ask them while you think of them. But we're going to request that the questions be addressed after presentations, but they will be taken down as you put them in, both the chat and Q&A. I think Q&A probably is a better way to put them in. Chat, you can chat with different individuals if you would like privately during this, but mostly if you would just have your questions down. We have a panel that will be put together, and we'll try to answer those questions in the end. Post that, I've actually would like to present some information on a EU COST program, and that's COST, all caps. It's a program for network of astatine research in Europe, in the European Union, and actually around the world, that they'd like to have a collaborative type of thing. And also maybe, hopefully time allowing again, that we can, I can present some slides that were prepared and given to me to present on the Japanese astatine production and sites that are going there. And I think it'd be kind of interesting to show what a couple of other fairly big programs in the world are going on. So what I'd like to do then today is to start off with a presentation of what's going on at Duke of Yutian Feng. Yutian, will you take it over at this point?
Yeah. Thank you, Scott. Can you hear me?
Yep, see you and hear you.
Okay. Thank you so much for the introduction, and thank you guys for giving me this opportunity to talk about the studies done at Duke involving developing targeted alpha radiotherapy agent using Astatine-211. Here at Duke, we produce Astatine-211 with our CS30 cyclotron and equipped with an internal target system. Our internal target has a rectangular shape. We coat the surface of the internal target with a very thin layer of bismuth, and we position this target at 28 MeV inside of the cyclotron. This internal target allows a higher current on the target and a bigger contact area for more production. This target cost has been very low and has been very reliable for the past 20 years as we have produced Astatine-211 in the past 20 years. Currently, we have an ongoing collaboration with Ionetics to upgrade our cyclotron and bring our facility to the 21st century. Yeah. So at Duke, we produce Astatine-211 on a weekly basis. The maximum amount of astatine produced here at Duke is 250 millicurie. We separate our astatine by dry distillation. So basically, we heat up the bismuth target and we trap the astatine that's released from the bismuth target. Recently, we have also distributed Astatine-211 to remote locations. The most recent one is to Johns Hopkins University for collaborative research. The transportation is about 6 hours. This table here includes the most recent 4 runs where we distributed Astatine-211 to Johns Hopkins. After a 3-hour radiation, we were able to produce over 150 millicuries of astatine, and we were able to ship out around 60 millicuries of astatine. Even after 6-hour transportation, we still have enough activity for clinical study. So in our group, we have been developing targeted alpha therapy agents using Astatine-211 for many years, long before I hop on the ship. In the recent years, we have gradually switched our research focus to small molecules, single domain antibody fragments, and noble metal nanoparticle platforms. These molecules are smaller compared to intact antibodies, so their pharmacokinetics are faster, and that is compatible with the half-life of Astatine-211. It is also compatible with intravenous administration. This is one of the examples for a past effort. Astatine-labeled full intact antibody 81C6 for glioblastoma. Patients diagnosed with glioblastoma have very poor prognosis with external beam radiation therapy. In this case, we directly inject the astatine-labeled antibody into the surgically created resection cavity during surgery. This will avoid the toxicity issue of astatine-labeled antibody in the bloodstream and in normal tissues. With the patient study, there's a clear benefit for patient treated with this treatment. Now, this is one of the more recent effort in our group, astatine-labeled PSMA targeted agents. Low molecular weight PSMA targeted agents is probably one of the most promising, one of the most popular concept in nuclear medicine, if not the most. Our first generation is developed in collaboration with the Martin Pomper group at Johns Hopkins University. This agent, DCA-BZL, has promising results in vivo. However, the kidney uptake and retention is fairly high. Keeping that in mind, the strategy is to develop the second generation PSMA targeted agent is to improve the normal tissue clearance, especially in kidney, salivary gland, and lacrimal gland. Simultaneously, we aim to develop blocker that's co-injected with the radiopharmaceutical to selectively lower the uptake in normal organs. Now, the second generation compound is VK290 lutetium compound. So the in vivo study has reviewed that the second generation compound has very good performance in vivo compared with the first generation. We have retained the high tumor uptake, even co-injected with the blocker, we were able to retain the high tumor uptake. And the clearance from normal tissues such as kidney, salivary gland, and lacrimal gland has been vastly improved, especially when co-injected with the blocker. It is also very important to note that the uptake of this compound in salivary gland and lacrimal gland is at a very low level. This is quite an advantage compared with other PSMA targeted agents, where toxicity in these organs are one of the biggest concerns. So we went ahead and did a therapy study with the second generation compound in ethamic mice bearing both PSMA positive and PSMA negative tumors. There's a clear anti-tumor efficacy observed with the PSMA positive tumor treated with our second-generation agent. Currently, we're optimizing the chemistry and radiochemistry for this agent to facilitate its clinical translation. Switching gears a little bit to another research focus in our group, radiohalogenated nanobodies. Nanobodies are single domain antibody fragments. They are an order of magnitude smaller than intact antibodies, about 13 kD, but they retain the nanomolar binding affinity of a full intact antibody. To label these nanobodies, we developed these residualizing agent that contains a residualizing group, a guanidine group here. This guanidine group can increase the intracellular trapping of radioactivity once the conjugate was internalized in cancer cells. It also decreased in vivo dehalogenation. And this pathetic agent can be labeled with F18 for PET imaging diagnosis, and radioiodine for beta therapy in Auger therapy, and Astatine-211 for targeted alpha therapy. To put this pathetic agent onto the nanobody, we have developed a variety of linkers. For site-specific labeling, we can use linkers that targets the cysteine residue on the nanobody. We can also use random labeling that targets the lysine residue on the nanobody. And there are also kit and linkers that specifically designed to improve the PK of the conjugate. So our labeling is done in two steps. First, we start with a tin precursor. We label the tin precursor with astatine-211. After purification and deprotection, we conjugate the pathetic agent onto the nanobody. Currently, the state-of-the-art agent in our group is iso-SAGMB. So we carried out a biodistribution study to investigate whether the substitution group of the guanidine group will affect the in vivo performance of the pathetic agent conjugate. So compared with the ISO version, this non-ISO version where the guanidine group is at 134 position, the ISO version has a higher tumor uptake in vivo. In normal tissues, a similar advantage was observed for iso-SAGMB. Compared with the non-ISO version, iso-SAGMB has lower uptake in thyroid and stomach. We selected thyroid and stomach to present here because these are the organs where astatine will go if it has fallen off of the conjugate. As you can see, a lower uptake in thyroid and stomach indicates a lower level of in vivo dehalogenation. In summary, we are capable of producing over 100 millicuries of astatine from a single run. In the past, we have distributed astatine to remote locations such as Johns Hopkins, and even after 6-hour transportation, there's still sufficient activity for clinical studies. I listed 6 hours here because that's what we have done, and that's also to demonstrate the feasibility. However, we can also distribute it to more faraway locations for studies. Our radiochemistry technology allows us to produce sufficient amount of product for clinical relevant dose. In the near future, we're going to translate these two agents to clinics. So in the past, our group has always been open to collaboration with people who are interested in astatine chemistry and using astatine produced at Duke. We have also hosted visiting scholars from Europe, from Asia, from many institutions within the United States. One of our mission is to help people learn the astatine chemistry and develop more research projects using astatine-211. Now, I'd like to acknowledge the radiopharmaceutical chemistry lab at Duke, my advisor, Michael Zalewski, Ganesan Vaidyanathan, our lab mates, Zheng Yuanzhou, Stephen Jannety, Darren McDougald, Joshua Guang, Rebecca Michel, and Sean Murphy, Michael Daly from the CYCLOTRON facility. I'd also like to acknowledge our founding agencies, Department of Energy and NIH. Here is a snapshot of a history. Thanks to, this picture is credited to Arne Freiberger, a program manager of Isotope Accelerator Facility. Thanks to the Duke administration in the 1980s, we were able to purchase the CS30 cyclotron and produce astatine, and it has been producing astatine reliably for the past 20 years. Thank you so much. I'd like to take any questions if you have.
In this virtual world, you don't get all the claps and whatnot, but Thank you very much, Yutian.
Thank you. Appreciate
There's
that.
always a lot of interesting stuff going at Duke, and very nice presentation. Thank you. Next, we'll wait until the end to do the Q&A, as I said, but you can put in those questions anytime so you don't forget about what your question was. I'd like to have Sherry Yanello give her presentation now.
All right, so I want to thank Scott for asking me to do this, and I really want to thank Ethan Balkin, who actually got us back into this. And I'm going to tell you a little bit about what is a new effort here really at Texas A&M, which is advancing research in Texas through experiments in medical isotope science. And this is an effort jointly with the Cyclotron Institute and the Nuclear Science and Engineering Center here at Texas A&M. Okay, so like I said, this started really when Ethan had the idea of pulling together universities that had accelerators and trying to form this network. So he called a bunch of us into Germantown in 2018 to talk about this. And out of that came a call for proposals. Last summer, we had a really very smart, industrious undergraduate who worked with us for the summer that started working on doing some bismuth experimentation to prepare for starting to do astatine separation. And then it was really just the fall of 2019 where we started to do irradiation to make Astatine-211. Some of the interesting initial chemistry has already been accepted for publication, and this really kind of puts us at a place where we are very excited to begin this project as part of the university network. So for those of you who've never been to Texas A&M, I invite you to come visit us anytime. But I thought I'd show you the layout of the facility. And we actually have two cyclotrons. One is a K500 superconducting cyclotron, and the other is the K150 cyclotron. And then you see a whole bunch of equipment that has nothing to do with astatine, because this facility has been here for over 50 years, basically with the mission to do discovery science in nuclear physics. And so I put together this a number of years ago where it really talks about how we do discovery science and workforce development. And then something that has happened in the last 20 years, which is realizing that we, beyond doing the basic nuclear physics, we had beam time available and started to do radiation effects studies in terms of chip testing. But moving forward, what we're going to do is change that radiation effects to applications as we take some of that cyclotron capability, the beam time that's available, and move it over into isotope production. So let me focus in on the K150, because that's the machine we use for astatine. It is an 88-inch cyclotron. It is a copy of the one from Berkeley. We feed it with both a negative ion source and an ECR source. So we can accelerate everything from protons up through fairly significant heavy ions. And this is just a smattering of the beams that have been extracted. For our purposes, we think of it as our high intensity machine. Of course, high is relative. But it's a variable energy machine, which is very nice. And it has a lot of capability. So it is used significantly in our science program, but we do have some time that we can use it for the production of Astatine-211. And what I wanted to do is blow up the footprint of the facility. And here you see that 150 cyclotron, the 88-inch. And here's the line you saw coming out of it to the analyzing magnet. And what happens is right here, we stick our astatine target, okay? And so this is where it is, which means that one of the things that we'll be doing as our project gets started here is working on target development, because we have perfected the art of melting bismuth targets. And now we need to work on the art of not melting bismuth targets until we're ready to. But we don't have a lot of space. I mean, there's a wall here. You can see the analyzing magnet goes around there, but this is where our target sits for the irradiation. Okay, so then once we irradiate and we pull the target out, we have some radiochemistry facilities. We do much of the work right now in a biosafety cabinet. We do have a hot cell in the radiochem lab that once we are able to get the beam intensity and the target integrity up so that we can make as much or maybe somewhere near the amount of bismuth as you just heard about from Duke, we'll have to move into the hot cell rather than the biosafety cabinet. Because of COVID-19, We have had to actually, in our last run, find another hood because of the de-densification of people per hood. And here you see Amy, that very smart undergraduate who worked with us last summer doing chemistry, has come back and she's getting ready to do some chemistry in a second hood. So we do have the capability of doing chemistry in 2 different locations right now. All right, so a little bit about some of that chemistry that we've done, and this is just a little chart. So we do our production, again, same thing, the alphas on bismuth to make the astatine. We do not do dry distillation. There was a previous effort at A&M to do dry distillation, and it was not super reliable. So we have gone to wet chemistry, and so we dissolve the target in nitric acid, and then we work on trying to separate out the astatine from the bismuth. And we've actually done a nice job so far. Here you can see our target. Again, it's an extended target because we run at the target mechanism at something like 10 to 15 degrees. So we take a round spot and disperse it over there. And you can see it then in our dissolution tube and then being dissolved. I want to show you one slide of some of the chemistry that we did in those first couple of runs. And this is looking at the astatine separation. So Amy last summer looked at what happens with bismuth and trying to extract out D values, particularly in the decanol. And you can see, this isn't her data, but this is astatine now. But what she saw was that the bismuth really doesn't come out into these things. But when you look at the astatine, here's diisopropyl ether and decanol, both people have used, and they have nice D values. They're not really showing a relationship with the nitric acid. If you switch over from an alcohol to a ketone, you can see you have significant more extraction of the astatine. So we ran a column where we took the column and impregnated it with octanone, and you can see that basically the bismuth comes through, the astatine sticks until we wash it out, and we get a very high purity and a high yield from that column. So we're very excited about that going forward. In terms of where we're headed and what our challenges are, because I'm hoping that some of the other panelists can help me here, beam is going to be a challenge. We need to work on intensity. As we work on intensity, we need to work on uniformity. As I told you, we're really good at melting targets, and hot spots in the beam mean you can take less intensity than if you can distribute it significantly. And we want to work on beam stability just to make sure that as we do an 8-hour irradiation, it stays constant and we can measure the integrated beam current. For the target, we need to work on the thermal contact and being able to withstand the beam without melting. We would like to get to a thinner bismuth target than we've successfully run with, because the excess bismuth just gets in the way of the chemistry. And we have to work on our fabrication and making sure we know the activity before dissolution. And as I said, our target mechanism right now requires manual removal of the target. And so we're going to work on some of those things as we develop a new target system. And then in terms of separation, what I've shown you is really some very nice first results of chemistry and separation, but we want to optimize that chemistry and then think about implementing it on a column. Okay, so while we make astatine here, I said, well, I don't just want to let it decay, so we formed some collaborations, and one is with some people over in the College of Veterinary Medicine to look at the treatment of osteoarthritis. We have another collaboration with some inorganic chemists in the chemistry department to look at some ligand exchange experiments, and with theoreticians to kind of understand the interactions through density functional theory calculations. And so all of that we think is a nice sort of broadening collaboration of people that are interested in using astatine to further the health. So my quick summary, I'm not sure how I'm doing on time, Scott, but
don't worry about it.
We've had 3 successful experiments so far where we were able to produce and recover the Astatine-211. And our early results on the chromatography really show high yield and high purity of the product. And we are working to build collaborations to facilitate the knowledge production and utilization of Astatine-211, really so it can be used as a medical isotope. So I want to show you, I have to acknowledge, this really has been a village of people that really are contributing pieces of their time, but they live in lots of other places. So lots of direct and indirect support for this project so far. And the Cyclotron Institute staff are amazing and they can't be thanked enough. They're really terrific. And also our rad safety people, just top notch and have been super, super helpful. So we could not do this project without them. And just to finish, as I said, COVID-19 kind of changes things, but for us, we said we're not going to let it stop us. So this was a picture taken in June for our last run of the team, all appropriately physically spaced down the hallway, so we didn't get together, and all masked. And that's the way we had to do our last run, just to be able to continue to do science in this COVID era. And just to acknowledge my whole team, the core team at the top, and then our collaborators below. And it really is a big joint effort. And I really, really want to thank Ethan for dragging us back into this as part of the university network. And to all of you who don't yet know how much you're going to help us as we develop our Astatine capability. So thank you.
Thank you, Sherry. Very nice presentation and a lot of stuff happening. I'm very happy to see that. Okay, so we're going to go on next to Mehran Moghbandi at the University of Pennsylvania.
So today I'm going to give a little update on some of the projects we're working on here at the University of Pennsylvania regarding Astatine-211. So one of the major projects that we have been pushing forward for the last several years is working on developing PARP-targeted alpha therapeutics using small molecule PARP inhibitors. And so what this slide is showing is how we're developing these agents as a treatment for high-risk neuroblastoma. We work in collaboration with the Children's Hospital of Philadelphia and the John Maris Research Group, who has really produced some phenomenal models that are able to recapitulate patient cancers and the actual disease they see in the clinic. So what they have developed is they take tumor samples from pediatric cancer patients who suffer from neuroblastoma, and they xenograph these tumors to a mouse. And these tumors actually never touch plastic, so they retain a lot of characteristics that actually are representative of the cancer. They further characterize their tumor models through genomic analysis with RNA-seq, and from this, you can actually identify novel drug targets and look at mutations that are associated with response. So what we do is we started working with the Maris lab to develop Astatine-211 radiotherapeutics. We first worked on Astatine-211 MABG, and later we started introducing this compound here called Astatine-211 MM4, which is a small molecule PARP inhibitor labeled with astatine here. And PARP1 is the primary target of this drug, and it's expressed solely in the nucleus of cancer cells, right on DNA. So it's a really incredible target for alpha therapeutics. And the potency of this compound is very impressive. So right now we've performed a lot of the chemistry development. Over here in this top right, you can see some of the patient-derived xenograph efficacy studies where we've shown that the maximum tolerated dose is sometimes curable for up to 15 weeks in these xenografts. These tumors actually did not come back, and you can see some tumors relapsing after 10 weeks or near that time. So out of these 5 patient xenograph models, we actually had a 70% response rate. So we've taken this data, we've done genetic analysis to look for mutations that would determine which patients would respond to this agent, Astatine-211 MM4, and we're really just gearing up to go into clinical trials. So this was an abstract presented by, This was an abstract presented by Juan Lee. The second abstract that was presented at the Society of Nuclear Medicine meeting from our astatine PARP group was evaluating PARP alpha therapy and checkpoint blockade in a syngeneic mouse model of glioblastoma. And one of the most impressive findings of this work was that the combination therapy in combination with the checkpoint inhibitor, an anti-PD-1 antibody, was that in combination with Astatine-211 MM4, you produced a longer progression-free interval, and we actually had 100% of these mice have their tumors cured, where anti-PD-1 or Astatine-211 MM4 had a faster progression and a lower cure rate. And some of the striking, sorry, my mouse is moving the screen on me. So some of the striking observations that we found, I can't do it. Some of the striking observations that we found in tumors that we analyzed by histopathology is when we looked for tumor-associated macrophages, tumors that were treated with anti-PD-1. Here's an example here where you can see macrophages have infiltrated the tumor, and there are a lot of macrophages, although there's not quite as many as with the alpha therapeutic alone. So what we think is going on is this alpha therapy is actually priming the innate immune response. And so in combination with PD-1 and an intact immune competent mouse model, you can actually get a better transfer from the innate immune response to the adaptive response. So we're going to continue working on this as we go forward. We've also taken this compound, Astatine-211 MM4, and looked at some preclinical toxicity. So one of the concerns with this agent that we first identified is PARP1 is highly expressed in bone marrow and some of the different blood cells, predominantly lymphocytes. So we performed a toxicity study in immunocompetent mice looking at 3 different escalating dose levels. And then we also use a fractionated dosing scheme. So we also wanted to look at after each dose, what toxicities were evident. And we analyzed these by colony formation assay, CBCs, IHC. And what we found is the bone marrow. So this is a colony formation assay where we're evaluating bone marrow progenitor cells. And the GEM is the earliest progenitor cell you can actually grow in a colony formation assay. What we found is we found a dose-dependent reduction in colony-forming units, which suggests that there is direct toxicity to this early progenitor, although we weren't able to completely ablate the bone marrow, which was interesting. And evaluating the same colony-forming unit after at different time points after just 2 doses of Astatine-211 MM4, we find the progenitor is reduced at 72 hours, although it increases to not statistically significant, but still lowered levels as compared from baseline. So we're thinking there's a rapid recovery. In lymphocytes, which is a little bit more, the results were more obvious. We saw a rapid reduction, and we did not see any dose-dependent effects here. So we think we're well above the concentrations or the activities where we would actually see dose-dependent effects. But even with the lymphocytes, so here is after 72 hours, you get a rapid reduction in lymphocytes, although they recover in 2 to 4 weeks. So another abstract that was presented at the Society of Nuclear Medicine meeting by Paul Martorano was the development of a new way to perform Astatine-211 dry distillation. And this is something that I was really excited about. And we've all been very excited about this. I think it was kind of a very creative project from the get-go, and Paul kind of just came to me with this idea, like, yeah, can we do the distillation a little bit differently? So what we designed is an induction heater that can actually, by pushing current through a copper wire, you can create a magnetic field, which then can create electrical currents in certain materials that are conductive and generate heat. So we're switching from the tube furnace to a method where we can directly heat our target. So here's what the induction heater looks like. This is a terrible design that was put together by our electronics shop. I think it's definitely the beta version, so it's much larger than we wanted. We have this large circuit and transformer, and so it's really just a proof of concept to begin with. But the business end is shown right here, where we have this small copper coil that has a insulator around it, so we don't electrocute or burn anything that we touch with this. And then the tube would just fit right inside here. So the results from this induction heater were that we were able to rapidly heat these targets, and I have a video here, so you can wait with me for the 1 and 30 seconds that it takes for this target to become glowing hot red. It's really, really awesome. And as you can see in this graph, the induction heater is able to rapidly distill astatine off of the target in less than 10 minutes. You can see that the target's already starting to glow, and as time goes, it'll become hot red, and that's when the target is really reaching the temperature of the melting point of aluminum. And compared to our old conduction tube furnace, which takes much longer, we were just able to have more reproducible results and faster distillations. And so this is something we're really excited about. And if you can imagine, you might be able to design small chemistry synthesis modules that incorporate a induction heater that can heat small coin targets, and it could be in a very small, a small design that could fit inside a hot cell. So the last thing that I wanted to just briefly mention is kind of where we're going with some of the astatine chemistry at Penn. We don't have anything to share with everyone at the moment, but really here are 2 studies that have made us kind of go back to the drawing board and start thinking about how astatine carbon bonds behave, or how small molecule astatine molecules behave in vivo. So this first paper came out of the group from Nantes, and they did some really nice calculations. I heard one of the earlier speakers was mentioning how they're starting to look at some density functional theorem calculations to determine the bond dissociation energy. So this paper was the kind of the first to go there, and I think it's a really useful tool that can allow us to evaluate astatine chemistry in silico before we have to produce it, distill it, and run chemical reactions at a really low quantity. So we have some work going on in this area to look at these methods for identifying stable astatine small molecules or stable structures. A second abstract poster that I wanted to bring up was from the groups out of Japan, and they radio labeled this neopentyl structure with alkyl halide bond or alkyl astatide, which is something that most people would cringe at in the field. And I think what they found is that this neopentyl structure most likely is not a substrate for cytochrome P450 or other oxidative enzymes. Something about these 2 hydroxyls most likely limits the metabolism, and it is only metabolized by phase 2 glucuronidation. And what they found is even with an alkyl astatide, this structure is stable in vivo. So it really makes you think maybe just avoiding enzymatic activity with any type of targeting vector labeled with astatine would be sufficient to improve in vivo stability. So we have some more work that's really focusing on these 2 areas, and we'll be able to share it soon. Lastly, we have been ramping up our production at Penn. We have a new target station for the production of Astatine-211 using metallic bismuth that was designed by Sumitomo Heavy Industries, and this new target system should get us up to producing at 30 megabecs per microamp hour with about 20 microamp currents. So we'll be at the point of producing roughly 20 millicuries an hour. Some other work that we did recently was looking at bismuth oxide as target materials. And although bismuth oxide, so the pros are bismuth oxide, it has a higher melting point than metallic bismuth. So we were able to irradiate targets up to 30 microamps, although because it is an oxide, it has oxygen, and the alpha 2N reaction with oxygen can create fluorine-18. So you have co-production of fluorine-18. And what we found is you really need to optimize the density of these targets in order to successfully have the stopping power to capture the cross-section of astatine and limit how much fluorine-18 you're going to co-produce. And then lastly, like I said, we have some new chemistry on the horizon, so hopefully everyone can just sit tight. And I'm really excited about all of the work really going on in the chemistry field of Astatine-211 at this time. So with that, I'd just like to thank the group at Penn and CHOP and all of our funding institutions. And I'll be happy to answer any questions during the Q&A.
Thank you, Mehran. Very nice presentation, very exciting work that you're doing. I guess the next presentation is actually going to be split between two people at the University of Washington. Well, I believe we start with Rob Emery, who's in our cyclotron facility there, and then we'll follow up with Yavin Lee, who is in the radio labeling part of this. So I'll let Rob take it over at this point.
Yeah, great. So yeah, my name is Rob Emery. I'm the director of the cyclotron facility at the University of Washington. And I'll just give you a bit of an overview of our facility and some of the targetry work we're doing. And then I'll turn it over to Dr. Lee, who will talk more to the radiochemistry and the application of the astatine. Great. So in the recent years, our medical cyclotron facility isotope production has come under DOE stewardship. And so with that, we get base funding that partially supports the operations costs. And then our radionuclide production comes under DOE guidance. So basically what we're making and who we're making it for, we're working hand in hand with the DOE to determine that. Our radiochemistry division is funded through base funding and R&D funding. Currently, we're providing Astatine-211 through the NIDC. We're also developing other radionuclides through the R&D funding. And the idea is that we'll be able to provide these other radionuclides once we achieve these production routes and we have the facilities adequately handle the higher levels of activity. Right now, we don't have a hot cell or automatic retrieval mechanism we can use. And that works fine for the astatine, but once we start looking into higher levels of astatine or other radionuclides, we're going to need some better handling facilities. So a little overview of our cyclotron facility. It's Scantronics MC50 installed in the early 80s. The thing that makes it unique is it's a variable particle, variable energy. We can do alphas and protons up to 50 MeV. Also do deuterons and helium-3, which is nice. The other great thing about it is we have relatively low downtime and high availability. So we're really kind of primed to start ramping up our radionuclide production program. Recently went through a facility upgrade. The original facility was mostly made for fast neutron therapy. Over the years, we've modified some of that. We're doing fast neutron therapy, proton therapy research, radiation effects testing, and then isotope production. You can see our original system. We added a 0 degree line down the middle, which is now where our astatine production target sits. And that's strictly used for our alpha beam producing either astatine so far or tin-117M, which we've done on occasion. But we're regularly producing astatine, every 1 to 2 weeks. And then down here on the right, this was originally our PET production station. It was sort of an afterthought add-on. And we turned that over to our target research station. With the upgrade, we've swapped out our switching magnets. So this gives us an extra 4 ports. But it also gives us a cleaner beam line, for example, down to our research station. It gave us a space to add in a new focusing quad to either focus the beam tighter to get more current on target or to spread it out to reduce the heat load. So it'll be a nice addition. We've also added quads upstream. Our alpha beam comes out pretty wide. from the machine. And with our original quads, the aperture wasn't big enough to capture that beam. So with these bigger quads, we can capture it, focus it, and get it down to the end of the target station. Just a couple of pictures of the recent installation. This was done at the first of the year. You can see on the left the new quads there in blue, and then the new switching magnet there being dropped in. If you ever have to go through this, we picked up over here on the left this laser tracker system, and we were able to drop this equipment in, over a long weekend and have it up and running in a few days. And we get sub-millimeter precision with this laser tracker in the beamline alignment. So it was really helpful to keep our downtime down. Here's sort of the final installation on the left. You can see over to the right the new beamline with the quads we installed. And then this picture over here on the right just shows us using some beam optics simulation software to quickly get that tuned. Again, just to sort of reduce downtime. We operate 4 days a week for fast neutron therapy, 52 weeks a year. So we really don't have an opportunity for any major downtime to make upgrades. Yeah, so that's it. Thank you. And I'll turn it over to Dr. Lee.
Good afternoon, I'm Yavin Lee, and I'm going to give the second half of the University of Washington update. So the amount of astatine produced and used at our site has increased over the years. We are producing astatine to support research in the automation of astatine isolation. We also provide astatine-labeled reagents to our collaborators to support preclinical and clinical research in developing radioimmunotherapy for blood-borne diseases. In addition to leukemia, lymphoma, and multiple myeloma, there are also research projects that are focused on targeting HIV-infected T cells and gene therapy. Over 2 curies of astatine was produced in the past 2 years. The demand for Astatine-211 increased dramatically after our first clinical trial started in 2017. We produced 457 millicuries in the first half of 2020. And we think by the end of the year, we might be at a similar level as last year. A quick update on the status of the astatine clinical studies. We are now supporting 2 clinical trials on astatine BCA-D10. The second one just started this year. 26 patients have been treated, and over 3.2 curies of astatine has been produced for patient treatment since 2017. We are currently applying a new IND for another astatine-labeled antibody. It is an anti-CD38 antibody for the treatment of multiple myeloma. In addition to clinical trials, there are a number of radiochemistry and preclinical research projects that use astatine. Our group is founded by the DOE to work on the automation of astatine isolation. Our collaborators, Drs. Green, Water, and Orozco, are funded by NIH and ASH to investigate new treatment for blood-borne diseases using astatine-labeled antibodies and hematopoietic cell transplantation. Astatine-labeled antibodies are being evaluated by the research groups of Dr. Steinmeier, Pincus, and Harrington as a potential method to target T cells and deplete latent HIV reservoirs. The research groups of Drs. Water, Kim, and Storb are studying gene therapy using Astatine-211 labeled antibodies and hematopoietic cell transplantation. Our group contribute in these projects by providing the astatine reagents needed. Finally, I would like to bring up the tellurium column method for astatine isolation. The details of the method have been published last year, and we would be happy to talk to anyone interested in this method. The isolation process is still being optimized. Our goal is to have no hydroxylamine hydrochloride and minimal or no residual tellurium in the final product. For the wet chemistry method using liquid-liquid extraction, the non-decay corrected yield of isolation for both the manual process and the automated process is about 60%. The tellurium column method can provide a yield of 90%. Currently, our semi-automated process takes about 1.5 hours from start to finish, which is about 1 hour shorter than both of the manual and automated wet chemistry process. This is all I have, and thank you for listening, and thank our funding agencies.
Thank you, Anne. That was very good. All right, so now we're going to switch over. We're going to have Graham Marshall from Global FIA. They have been involved in automation processes, and he'll talk a little bit about some of the things that they're dealing with there.
Well, first of all, thank you, Scott, for the opportunity to present our work and to do so at a late stage in the organization of this group. I'll dive right in. Pacific Northwest National Laboratory and Global FIA are collaborating in a phase 2 SBIR project to facilitate regional availability of astatine in the US, specifically with regard to the purification of the astatine once it has been produced. 6 process unit operation comprising dissolution, acid conversion, solid phase extraction and neutralization. And this is followed by a choice of labeling or dewatering of the neutralized astatine-bearing salt solution. And these are being optimized and integrated into a compact advanced development model for automated solution handling. This model, once tested, will be deployed to 1 or more user facilities to demonstrate operation in an end user environment. Progress has been made with several unit operations, and a CAN bus-based distributed control system software is in the process of being developed, as well as a series of sensors to monitor the operations. And if you have any questions on our work, by the way, my email, and Matt O'Hara, my colleague in this project, is given under our organizational logos. With limited access to facilities that we could, where we could work on Astatine-211, our work to date in the project has focused on the development of our distributed control system and the supporting sensors and unit operations where it was not important to have the astatine present at this stage. We have developed and are testing a useful toolbox of sensors that will be included in the final fluid handling system. And these include a conductivity sensor for reagent leak detection, a radiation sensor for monitoring the transport of the astatine through the system, and for isolating the product-bearing eluent in the solid phase extraction unit operation. As the astatine is eluted from the column, as shown in the first graph, the sensor controls a valve which gates and isolates the eluted product from the bismuth waste and wash solution. The graph shown there was actually in one that we did in collaboration with Dr. Lee at University of Washington, eluting from her tellurium column. Pressure sensor is used to monitor fluid flow and back pressure in unit operations. And the middle graph shows the pressure profile during target dissolution and indicates when the target is finally dissolved. A vapor sensor has been developed to track the dewatering unit operation and may be used in the acid conversion step as well. We've also developed temperature sensors and controllers for our heaters, condensers, and temperature sensitive unit operations. These will be used in several of the unit operations. A pH sensor is used in the neutralization unit operation to track the pH during neutralization. These smart sensors and fluid manipulation devices, such as pumps and valves, are all controlled via CAN bus by the distributed control system software, which is under development. Data obtained from these sensors, such as the radiation sensor, the pressure sensor, and the humidity sensor that we've depicted here, have already been put to good use in unit operations to study and optimize these unit operations. And finally, in closing, I just want to acknowledge the excellent work of my colleague at Pacific Northwest Lab, Matt O'Hara, and funding from Department of Energy through Ethan Balkan and Michelle Shin. Appreciate their contribution to this work. Thanks, Scott.
Thank you, Graham. Very nice presentation, very encouraging. Okay, so at this point, I think what I had in mind is that we want to make sure that everybody has an opportunity to ask questions and get those taken care of. I'm still hoping that we have time for me to present some slides that were sent to me about the cost program and the program of Astatine in Japan. But I would like to make sure that all the people that are online here, if you have questions, that we get those questions answered before we go to a couple of presentations, informative presentations. Okay, so I'm not sure, Karen, how do you want to handle the things? Let me just say that we have put together a moderated session of panelists that include Mike Zalitsky from Duke, Sherry Yanello from Texas A&M, Bob Mock from the University, about our opinions, our knowledge in the area with regards to astatine production or research that's more broadly in scope. I would hope you might address them to our panel. From that point, as I said, Karen, I'm not sure exactly how we want to take this forward. So do you have the questions at this point?
So can you hear me okay?
I can.
Okay, so there is a Q&A segment down on your bottom bar. If you click on that, you can see the questions that have already been asked. There's been 5 questions.
I know, but
okay.
So
most of them, it looks like, have already been answered by Maren.
Okay.
So if any of the other panelists or presenters would like to elaborate, that would be fine. Or
okay, then I will open it up if there's not any, and ask if any of the attendees, virtual attendees, have any broader questions about astatine production or astatine research going forward. Yeah. Okay, I see some comment. So one of the comments I see from Matt O'Hara is his comment, wow, 1/1000th the current of bismuth mass. That would be amazing. Matt, I'm not sure exactly which one you're talking about here as far as the bismuth mass.
There's another question on the answered page about the thickness of our 90-degree target. And so he was asking me what the mass was going to be.
Oh, okay. I'm sorry. I missed that.
Yeah, and so with this 90-degree target, we're looking at a beam spot size of about 18 millimeters in diameter. And with the smaller, with that smaller target, so this is getting away from that longer angled incident target. That and the fact that we're going thinner without being fully stopping, it really gains us a lot in heat transfer. And with the astatine cross section, we can really, get a lot of the cross section without having to have all that heat dissipation in the bismuth. And so that's what we're looking at.
The 5 milligrams sounds really low to me, Rob. We should double check on that number.
Yeah, definitely
Yeah.
check my math.
Yeah, I agree with you.
But yeah, the target's going to be quite a bit thinner and quite a bit smaller than what we've been working with in the past.
Okay, so one of the questions that I have here is, what's the fourth university center of astatine production? Well, the ones that we had today are Duke University. Duke has done this for a long time and really led the way in production. And as you could see from UTI, they produce a lot of it. Sherry Yanello now at Texas A&M is producing it. She wants to produce a lot, and hopefully she'll be able to get there. So that's the second one. The third one is University of Pennsylvania and now University of Washington. So those are the four sites that are currently working on this and in the U.S.
Hi, Scott. I saw a question asked from Gabriel for
Yes.
the target thickness from our production. So our internal target has a thickness for the bismuth layer about 100 micrometer, and we are actually trying to optimize that thickness. And there's another question that's about the minimum activity for medical studies. So we've had the luxury of producing a large amount of astatine for our studies, so we never really had to worry about this issue. I think this comes down to the question of what kind of chemistry that you're using. For example, in our chemistry, we use a tin precursor, so you can use fairly low amount of activity to start with, sub-millicuries maybe. And for cell studies and animal studies, with astatine as an alpha emitter, you really don't need much activity to start with. So, but Scott and Yawen at University of Washington and Marion at UPenn, they all have very different chemistry from ours. So yeah, the chemistry, I guess, it's the answer to this question.
Yeah, I mean, I guess I would say that it's more the targeting agent than the chemistry, in the sense that if you have a small molecule that is released very quickly out of the blood, you may have to have more activity to get a certain dose to the cancer. On the other hand, with antibodies, in our case, where we're targeting a fairly large number of cells within the blood system, we're up on the order of, I think we've had some patients with 30 to 40 millicuries delivered to the patient dose. But some other ones where you have a lot fewer receptors, a lot fewer things. So I think we're probably on the upper end of what you would need. A receptor binding where you've got a very small number is not going to take much. But in the case where we're targeting a lot of cells within the blood, we are having to put in as much as 30, 40 millicuries. And the time it takes to make all that, do all the QA, and we have a particular issue with our antibody in that we have to infuse it slowly because it has a reactivity by itself. And so we're having to start with over 100 millicuries to get, let's say, 30 millicuries injected, and 100 millicuries produced to get something like 30 millicuries injected. And so, I don't know what the minimum would be, but I think that's got to be getting close to the upper end of what one might have to have. Okay, there is a question here from Hushan Yan. Here is the shipment statement. Can you talk more detail? Purification, approximately 14 millicurie material will be shipped as bismuth nitrate salt, and material will be shipped overnight. Significant decay, approximately 1 to 2 millicuries at receipt. I mean, the unfortunate thing is, if you're at Penn or some other place, Duke, where you can have large area or number of facilities around institutions that can use it, you might be able to get a good amount shipped. Where we have to ship out from FedEx to, Middle America, Eastern US, we anticipate only 10% getting there because we figure it'll take 24 hours. And in 24 hours, there's only 10%. So that really is the bottom line as far as the current situation. Now, those of you at Johns Hopkins and some other places that are close enough to be delivered to, that number can be larger. And so you don't have to ship out as much for them to receive it. And again, so let's see, again from Jan, do you receive more production sites to build up? I assume what he's asking is, do we see more sites in the university network, or will other places? And I don't believe I can answer that question. I don't know if anybody else has an idea. Ethan may be online. He may have an idea about that. Ethan, are you online?
Ethan is typing in the chat. There are more sites, Scott, going from west to east: UW Seattle, UC Davis, TAMU, Duke, and Penn.
Yeah.
I have been unmuted.
Oh, very good.
Welcome.
Thanks, Karen.
Show yourself.
I don't know that that's possible. Yeah,
okay, no, that's fine.
But I'm the disembodied voice at this point. Yeah, so at this point we have 5 university sites that are involved with what is largely an R&D effort for the majority of sites, with the exception of University of Washington and Duke. The rest of the sites that Scott just listed off, or I'm sorry, that Sherry just listed off, are really undergoing what's an extended R&D process. That is to establish a capability and then to ascertain whether or not they can meet particular levels of production that would make distribution regionally a viable effort. And so that's the R&D effort that's being undertaken right now. This utilizes, as best as I've been able to determine right now, all of the available cyclotrons at universities that can accelerate an alpha particle. What does this mean? This means that if other universities choose to suddenly install alpha emitting or alpha accelerating cyclotrons, would we consider adding them to the network? We would certainly give it a thought. We have not at this point closed anything down, made any final determinations. It's like I said, it's largely R&D and seeing how much coverage we can get for the country. We fully recognize that in order to realize full utility of astatine, or clinical utility and adoption of astatine at some point, the only way that this is really going to get going in a big enough way is for industry to get involved. And so that's, I think, where I want to leave it.
Okay, thank you for your input. Getting some more questions here. Any comments about solid tumor application? I would just comment myself, and then I'll have some other people comment. One of the issues, we've studied this a little bit in our studies, and at least with intact antibodies with astatine on it, as you would expect, we don't get very good penetration, and therefore we don't have good tumor control with that. On the other hand, I think that if you look at the issue of small molecules and fast penetration, it's very possible that you could use this for solid tumor. I've always looked at these alphas as trying to meet the real unmet need in cancer therapy, and that is metastatic disease. Now, that isn't to say you can't do cancer therapy, and certainly the PARP results with maybe activating immune systems and things like that may be really important in the future. But my own impression is there's a big need. If nothing else, there's a huge need to try to treat metastatic disease in these cancers, because that's really not changed a lot in many, many years on treatment. So that's my answer. I open that up to other people. If you would raise your thumb or whatever you do, I think that Karen can have your voice heard there.
Hi, Scott. Let me address this question from a Duke standpoint. So the two of the cases that I listed in my presentation, one is PSMA agent, PSMA low molecular weight PSMA targeted agent. This is done in collaboration with Martin Pomper group at Johns Hopkins. We do see a very good tumor penetration in our recent published study using alpha camera to see whether the activity has reached a deep level in the tumor. We have had some very promising results. On the other hand, with our Astatine-211 labeled nanobody, and because of the small size, it does have a better penetration than intact antibodies. So other than just be on the surface of a solid tumor, it does penetrate well. So I hope this answers your question.
Okay. Other people want to answer or discuss this point?
Yeah, so I'll just comment really quickly.
Okay.
With our small molecule PARP inhibitor, we get great tumor penetration, and we're actually able to effectively reduce bulk tumors in mouse xenograft models. And this is something we're still trying to wrap our heads around. It wasn't the obvious thing, but we've been able to show, even in studies where the tumor has recurred and it grows to be about 1 cubic centimeter, we can reduce that all the way down to a non-measurable level. And so I think small molecules definitely get good tumor penetration and could be used in solid tumors.
Thank you. Okay, so there's a couple other questions here. Any astatine evaporation on business target irradiation? I think we have looked for that in our own case. We have pumps that have charcoal filters on them. There seems to be a very small amount. I think that when it forms, when you get to nuclear transformation, it tends to be encased in the bismuth. And it even takes 700 degrees to get it to come out. And so we haven't seen that. We have not seen, I mean, there may be some sputtering. We like I said, we see a little bit, but not much. So my comment is I don't believe that there is much of that happening. Okay. Ionetics Corporation is planning to commercialize site in Lansing, Michigan. That's great. I like to hear that. And so thank you for sharing that with us. Okay, are there other questions at this point?
Scott, I think one question was missed from UMA doctors. I can't find the question now.
Okay.
That was a question asking which site is the fourth site.
I answered that one.
Oh, you also, I'm sorry. Yeah.
Yeah. Okay, so comment on monitoring methods, since I have not, and Astatine is really quite nice. And the fact that it only has some low energy X-rays. And in fact, with Sherry showing, when we get up to these high quantities, we just use a glove box. It isn't, we still, try to protect people, but the reality is that there's not a lot of emissions from pure astatine when you make it. And so handling it has not been a big problem for us from dosimetry or from doses, personnel doses. You want to be as safe as you can be, but it is not difficult to handle, unlike a lot of the other betas or high energy photon type of emitting ones. So I'm hoping at this point that I can move on and present for the EU cost program some slides that Professor Justin sent to me for me to present. And then I hope to follow that up with the story of the Japanese efforts that are going on that Koshin Washiyama sent to me. So I'm going to share my, I'm going to share the screen here with the cost program at this point. There's a cost action in the European Union to have a network for optimized astatine labeled radiopharmaceuticals. Next slide, please. And so what is COST? It's an intergovernmental program created in 1971 dedicated to financing European and international research networks, partnership agreement with the European Commission, EU funded in addition to member, and management of costs associated based in Brussels. Next slide, please. Okay, so went the wrong way. Let's go the other way. All right, so the cost, and I'm putting these up because Professor Justin said, well, you need to show these. I think the cost action has different things that are associated with it. Let's go to the next slide. One of the things that they're trying to do is have an inclusion of so-called less efficient European countries. And I assume, you know, some that aren't in the EU. Let's go to the next slide. And they're open to international cooperation. Here again, just a sharing of information around the world with people that are working with astatine. Next slide, please. Okay, so cost financing, about 120,000 euros per year, mainly for academic private association. This is a project for 4 years. Network activities, not funding equipment or research, meetings, workshops, and conferences, short stay missions, training schools, and dissemination actions, including publications and open access. Next slide, please. Okay, again, this is sort of saying what it said before, but there's more information on this. Next slide, please. Okay, state of the art. Astatine is considered one of the most promising radio elements for targeted alpha therapy. Some people would think that's true, and maybe some other people would not, but I believe it's true. There is very little production, very few production centers in the EU capable of producing it. That's true in the US as well. Few research centers use Astatine-211, which has always had a problem with developing things. It's necessary to have a different mode of deployment, external hydrotherapy, hydrotherapy. So basically, setting up a network where you can have centers for this production and probably treatment. Different radio labeling techniques have been developed but remain improvable. I'm not sure exactly what that means. I'd have to talk to him about that. But we need to develop more, and I think the Marin's comment about the development of highly hindered ones seems to be interesting. Automation of these radio for clinical trials must be optimized. Okay, next slide, please. Manufacturers need to ensure the development of their products and requires the need of critical mass of user. Hospital practice limit development. Regulation of clinical trials limits the achievement of these with a certain EU disparity. Targeted therapies are expensive. Yes, that's true, but if they're effective, it's going to work. Patients do not always have access to information. Distrust for radioactivity. I might add even more distrust for radioactivity that are alpha emitters. Next slide, please. Okay, so this is sort of the areas that they've identified that need improvement and that should share that information. Identification networking of production centers, exchange of experience of high activity production processes, particularly QCs, GMP, radio protection. Exchange of experience on radio labeling and automation techniques, innovation for the design of more stable ligands, which I truly agree with. Creation of a network of patient injection centers. So treatment centers, centers of excellence, whatever you want to call them. I think this is, may or may not have to be. I mean, certainly if one looks at the potential for radon as a parent, one could say that you might have a different situation. But reality is that initially that's what we're going to probably have to have. EU and national law regulatory formation of patients association and nuclear health education. Next slide, please. Okay, so objectives. We get now to the objective. Bring together forces at the European level to be able to effectively address the different issues, to identify pathology in which Astatine-211 will be relevant, particularly in looking at dosimetry and radiolabeling aspects. Long-term goal of this project is make astatine targeted alpha therapy available to all citizens to improve healthcare and quality of life. I think we all would like that. Okay, let's go to the next slide. Choice of setting up a cost action should allow the creation of a network of producers, researchers, and users through participation in events, workshop, conferences, short-term scientific missions. First step will help structure an Alpha EU network. First step of response to a Horizon Europe call, and I'm not sure I know exactly what that is, but duration 4 years, and deadline for this was in September 2019. It was accepted in March of this year and will be started in October 2020. So I think this is great. The next slide, please. This will be kicked off this fall. Participating countries, you can see the list here. I'm not going to read those off. There's cost members, there's neighbors, and then international cooperation. I know they've reached out to me and some other people in the US that will be involved. I think Mike Zalitsky and Matt O'Hara were ones that I know about. Okay, next slide, please. Kick off at the end of October 2020, validation of the working groups and nomination of the leaders, working group leaders, time, schedule, budget, open to any new partnership from academia industry worldwide. So, and here is Professor Justin's email address. I want to thank him very much for sending these. I think it's nice to be aware of other activities in astatine, not just in the US, but what's going on for other ones. Now, if I could get up the slides that Koshin had sent, I'd appreciate that. Okay, so Japan seems to be quite active in the area of astatine research and production. Koshin Washiyama is from Fukushima Medical University. And I don't know if you can go full screen on that or not. There we go. Thank you much. All right. So next slide, please. All right. Astatine available in Japan. That's great. So there's the Research Center of Nuclear Physics at Osaka, Takasaki Ion Accelerator, Quantum Medical Science Directorate, Nishan Center for Accelerator Based Science, Advanced Clinical Research, and Tandem Accelerator. Oops, let's go back to that slide if possible. And so there are different things that they're doing. He noted here that Alpha 2N, of course, is the primary one, but the tandem one is looking at Radon-211, looking at both dry distillation and wet chemistry. And he's given some date and some people that are involved in it. I don't want to take too much time to go through all of this. The slides should be available after this presentation. Next slide, please. So there are 5 sites that produce astatine, which we listed in the last slide. And it shows, you know, their distribution with the slide shows their distribution within Japan. And as I said, there was one site that's producing the Radon-211. Interestingly, they have 13 sites. It says more than 13 sites that are doing chemistry or involved in astatine research. Next slide, please. And I guess this is just the same thing with, I guess, transiting from one site to another. Next slide, please. Okay, the short-lived RI supply platform. This program will provide stable supply of research radioisotopes throughout the year and technical support for safe handling. Supply of short-life isotopes that cannot be purchased from commercial base. A prompt and stable supply of the world's highest level accelerator facility association and support for promotion of basic research, wide range of fields. I would have to say I'm sorry that I am not more in tune with what's on these slides, but I just received them this morning. So what he's highlighted here is astatine being involved in that. The next slide, please. Middle-sized cyclotron, so there's MP30 at Fukushima, and he lists some different properties that they can have: a deuteron beam, alpha beam, which is required, of course, and estimated production in megabecquerels per microamp hour. And next slide, please. Okay, I think this is an interesting slide in the sense that, well, for one thing, it's put it in millicuries, which makes it a little easier for me. It shows the sort of monthly production of astatine at Fukushima Medical University. It does show that produces quite a bit now. So I anticipate a lot of research coming out of this effort. Next slide, please. In summary, before 2010, Japan had only one facility for producing Astatine-211. Owing to the advent of radium, Sofigo, with the efficacy to prolong the overall survival, they changed. So it's accepted after that, I guess, an interest in using alpha emitters. I think that has helped in the US as well. Due to the availability of target materials and the usability of cyclotron, reduced astatine was considered the most appropriate candidate to make. 5 facilities, as I said before, there are just one facility for radon. Short half-life, astatine-related chemistry, restricted to production site in its vicinity. Again, a regional type of production, if you will, and probably centers of excellence for treatment. Platform has been launched, availability of astatine has improved. Okay, so unfortunately, like I said, I have not had a lot of input on the slides, and I did not have a chance to really look at these much. I think it's always interesting to see what research is going on, what production is going on in the area of astatine in other parts of the world. So I appreciate very much Professor Justin and Koshin sending these slides so that I can at least introduce things to you about those. Okay, so I think we've pretty much run our time, you know, and I really hope that those people that have joined us on this virtual meeting got information out of it that will be useful to them. So with that said, I think we probably will close the session, and I thank all the speakers. We didn't have much of a panel, but I thank the panelists as well. I thank all of you for your interest in astatine, and I hope we can continue to show progress in making it more available and understanding the chemistry and its uses more in future years, and I hope to see you again at user meetings in future. So with that, I think we'll close this. Like I said, I hope all of you got some information from it. Thank you all.